Be talking to you about large species of heteropods found in the northern Gulf of Mexico. Today I'm going to share with you a little bit about the background information on the families and the species from my study, and details about the gateway research programs that allowed this research to come to life. I'll also share the methods of how we obtained our heteropods and what we did with them, and then I'll go through the results that I reached and summarize this information and share a little bit about the importance of the study. Lastly, I have a few acknowledgements to toss out there for those who were instrumental in my education, and then ask if anyone has any questions. So what did we want to know about these heteropods in the Northern Gulf? First, we asked what are the spatial distributions of the 5 large species of heteropods found in the northern Gulf of Mexico. And secondly, we wanted to know what are the vertical distributions and are there diel migration patterns for these species. Thirdly, are there ontogenic growth patterns in heteropod depth distributions? Are the eyes of these vertically migrating species larger than the non-migrating species? And lastly, do heteropod eyes develop at the same rate in relation to their body length or body growth, or at a varied rate? Let's begin with a little of the background information, starting with the classification of heteropods. So Heteropoda are commonly called sea elephants. They're from the phylum Mollusca and the class of Gastropoda, in the subclass of Caenagastropoda, in the order of Morpha. And finally, the superfamily that they're all held in is Pterotracheidae, which contains 35 different species recognized worldwide. I focused on the large species, which is 2 families, Carinariidae and Pterotracheidae, and there are 2 species within the Carinariids and 3 species of the Pterotracheids. The third group, the third family, was excluded from my study of Atlantids. So from this image, it's kind of easy to see why they're called sea elephants with that nickname. There are 3 main body segments to heteropods. The first is the proboscis in the head region, the main body, and then the tail. The proboscis contains radula, and they're unique in morphology among the different species. And these radula can protrude out of the buccal mass to grasp a hold of prey. The head region also contains large complex eyes that are pedestaled on stalk-like projections, and they terminate in a spherical cup that contains a lens with a short focal length. The main body portion is mostly transparent. It's long and cylindrical, tube-like, and it contains a swim fin. And the swim fin is an evolved modified terrestrial snail foot. The fin is positioned, interestingly, on the ventral side of the animal. These animals swim inverted, looking upward for hunting purposes. The last segment of the body flattens and fans out. It's the tail. And at the junction of the tail and body is their visceral mass. And this contains their digestive workings in the gills and the reproductive anatomy. So that's a little bit about them generally, anatomically. So what are the differences between the families and the species themselves? So the first family, the Carinariidae, they're generally much smaller than the Pterotracheids, and they have a short, stout proboscis. The main feature that differentiates these 2 families is the existence or non-existence of a shell. The Carinariids have a shell, and there's a couple different types. The first type is a partial shell, which is embedded in the dorsal tissues of the visceral nucleus. It's kind of like a thin fingernail-like covering. That's in image A up there. And then the image B is the second type. It's a keel-shaped covering, and the visceral mass of these types of Carinariids hang kind of low off a stalk, and that shell surrounds them like a small canoe. The second family, the Pterotracheidae, they don't have any shell at all, and they're much larger. They have long tubular bodies and very long flexible proboscis. These species are more morphologically separated by a couple of differences: their visceral shape and their eye shape. In Coronata, their viscera is long and narrow and very pointed, and in Hippocampus and Scutata, it's very short and thick and teardrop shaped. The eye shape in Coronata and Scutata is a tubular shape, where the retinal base is virtually the same width as the other end where the spherical cup is. And then the other eye type, which is found in the Carinariids, Placenta and Lamarcki, as well as Pterotracheidae hippocampus, is an asymmetric shaped eye, and this is where the retinal base is much wider than compared to the narrow spherical cup. And what makes heteropod eyes so unique is the way that they view the world around them. These different species have different length retinas that's in that retinal base. Generally, this retina is a few receptors wide, such as 3 to 6, depending on the species, and then several hundred long. And some species have straight retinas, other ones have ones that are curved, like in a horseshoe shape. And the way that they visualize, what they do is they scan their environment of their close proximity. They can most effectively scan when they're not swimming. The different species, they have varying timing and their cadence for the way that they sweep their eyes through their field of vision. So what they do is they sweep up and down through a certain degree of, let's say, 100 degrees for their field of vision. It can be different for different species. The image is transmitted to the retina, and then they take off in a swimming bout for a few seconds, and then they stop again and do the whole process over and over. And that's pretty much the life of heteropods, looking for something to eat in this method. So that's a little bit of the background basics on heteros and some of the details that will familiarize you, with the study. Let's transition and talk briefly about the research programs that allowed this research to come about. The first program is called the Offshore Nekton and Sampling Analysis, known as ONSAP. And then the second program that I'll speak on is called Deep Pelagic Nekton Dynamics of the Northern Gulf of Mexico, known as DEEP-END. So that first program, ONSAP, it was one of the natural resource damage assessment programs, a NERDA program through NOAA, that evaluated the immediate impacts from the 2010 Deepwater Horizon oil spill. And its purpose was to enhance our basic knowledge of the biodiversity and abundance and distribution of deepwater fauna in the Gulf. And this sample set that came about from ONSAP is one of the largest of its kind that's ever been collected. DEEP-END was a midwater field program, and it was supported by GOMRI. Its focus was to develop a quantitative, taxonomical, comprehensive assessment of deep pelagic fauna for the Northern Gulf again, and specifically the region right around where the spill occurred. This includes an examination of long-term consequences of that oil spill on those faunal assemblages. And the data that was collected there gives researchers the ability to establish a time series that can detect ecosystem shifts or responses. Both of these programs took a lot of manpower and a lot of laborious hours to retrieve all of the specimens and create the databases. This whole process started out at sea. So these crew series were on board the RV Meg Scansey for ONSAP and the RV Point Surf for DEEP-END. And the heteropods that were sent to me were collected by those who did all the heavy lifting out at sea by collecting the preliminary data, doing all the trawl work, washing down the nets, separating the taxa, packing it all up with all of their notes, and then shipping it out to the respective labs for the different proposed projects for the programs. The samples were collected from a total of 46 different sampling stations that covered a 420 by 120 nautical mile grid, and we sampled from the surface down to 1,500 meters deep. The circles, the red stars on the map indicate all of the deep end stations, and all of the circles in white, gray, black, as well as underneath all of those red asterisks indicate all of the ONSAP stations. The ONSAP cruises that provided me my heteropods, there were 3 in total in 2011. They were 4 weeks long each cruise, and then deep end, there was a total of 6 cruises through 2015 to 2018. The ones that I used for my study were 2015 to 2017, and there were 5 of those cruises. And those were 2 weeks long. And the sample gear that we used through both programs was discrete depth trawl gear for biological sampling. We used a 10-meter square mock nest midwater trawl. That's a multi-opening and closing net and environmental sensing system. That system was used for all heteropod collections and a whole bunch of other taxa as well. And the sample specimens used from this study were quantified by trawl volumes filtered with reliable net deployment data that confirmed the accuracy of all of our sample depths. So once I received all the specimens into our lab, I did the laboratory work by identifying the species, recording all the measurements of interest by way of microscope and digital calipers, scales, and dissecting tools. For all of the very small juveniles or any substantially damaged specimens, I identified to the lowest taxonomical tier possible, and I entered the species and all of the measurements into our database, and then I began the really exciting process of using that data to answer my hypotheses. So for a minute, let's talk about how many that I measured and identified, and then we'll hit those research questions. We collected almost 3,500 heteropods, and we identified all of those and measured them, databased them. And out of those 3,500, 1,993 were quantified samples, and these were used for the detailed comprehension for the study. All of the almost 3,500 samples were used for the spatial distribution descriptions, and the 1,993 quantified samples were used to analyze the vertical distribution, Diel migration patterns, and ontogenic growth patterns. And out of those, we pulled 333 quantified samples that came from deep end specifically, and they were used for the eye size comparison to depth portion of my study. So let's go back to that very first research question that I had. What were the spatial distributions of 5 large heteropods in the northern Gulf of Mexico? So I created spatial distribution maps. I plotted using ArcGIS on top of maps of the northern Gulf of Mexico. And overall, the heteropod abundances in this region were highest along the upper continental slope and lower along the deeper slope and the edges of the abyssal plain. And our highest quantities of heteropods were found along the DeSoto and Mississippi Canyon regions. However, the patterns did vary among each species. There was a relationship of the canyons and slope distributions. It was more clear for these 2 species. In red, we have placenta, and over on the left in blue is coronata. Also to note, coronata was also the species that was found at every single sampling station, and this was by far the highest collected specimen species through both programs. Overall, heteropod counts were greatest in 2011 compared to 2015 through 2017. I compared the summer cruises for each program, and ONSAP in 2011, we collected over 2,600 animals. And for 2015 through 2017 on deep end, we collected much lower at 440 total. So I did a t-test to check the statistical significance between these capture rates. The p-value was greater than 0.05, so the difference between these samples sets was not statistically significant. The reason that we deemed was because the trawl volume filter was much higher during ONSAP and less for deep end. If you remember, those cruises were longer for ONSAP, 4-week cruises, and the deep end cruises were 2 weeks. And just the overall filtering was greater for ONSAP as well. So this pattern of abundance, it's reinforced by this map. It's a chlorophyll map showing all the chlorophyll in red, orange, green, and yellow, and then that blue portion is the intrusion of the loop current. This is from a deep end report from our second cruise. The main prey of heteropods is copepod crustaceans and different zooplankton and larvae. And this region of the Mississippi plume shows some of the highest zooplankton abundances for the entire Gulf of Mexico. And these heteropod distributions are supported by similar patterns that people have done in previous studies on phytoplankton and zooplankton abundances, and they do correspond in a similar way to the loop current as well. I'd like to note here how heteropods are typically described as occupants of the world's tropical and subtropical waters. However, there was a study recently in 2014 by Wall Palmer et al., and he stated that heteropods actually show evidence that they are thriving in colder waters, such as south of Australia, and definitely past geological records show them for different glacial periods in cold waters. And this is starting to pose a challenge to those latitudes that are typically associated with heteropods. So not only did we want to know what they were doing on the horizontal plane, we also wanted to know what they were up to vertically. This leads us to that second research question of what are the vertical distributions and diel migration patterns for these heteropods. So the heteropods were mainly found in the epipelagic and the upper mesopelagic depths of 600 meters and higher. We found throughout, though, the whole measured water column, we did find them throughout, all the way down to the last depth range that we measured, which was 1,200 to 1,500 meters. There were 2 species of greatest abundance for the maximum measured depth, which were Lamarcki in that second set of columns and Carinariidae in the third. And just to note, there is something that these 2 species have in common. They're both the largest in-body species within their 2 family groups. So next I compared the abundance of each species by depth and separated them by day and night catches. I used RStudio to make these depth distribution plots that helped isolate possible migration. Lamarcki placenta and hippocampus didn't show evidence of diel migration, therefore I deemed them non-migrators. However, when we looked at the Coronata and Scutata plots, there was evidence of diel migration. The large white box for the daytime of 200 to 600 meters dissipates for its nighttime counterpart across from it. And then the large black depth box for nighttime at 0 to 200 meters has gained heteropod population, which seems to be the apparent movement from that daytime depth range up at night. These pterotracheids, Coronata and Scutata, they are the largest bodied and strongest swimmers from all the other heteropod species. And these advantages may be allowing them the ability to have greater migration capabilities and more success in feeding and predator avoidance. Vertical migrations, they're a current major focus among a lot of research. Just to point out a few, Jessica Bonicelli et al. Did a research published in 2016, vertical migrations of gastropod larvae, and Thor Clevier in 2016 also did a study on contributors to the scattering layer, and Sergio Bonaker in 2018 studied copepod vertical migrations. Most of the published research that has been done on heteropods has involved mainly the smaller heteropod family that's not a part of this study, the Atlantids, and they've also been mainly out of the Indo-Pacific region. And these studies also only went as deep as 600 meters. This study in the Northern Gulf that we've done is the first deep water study on pelagic snails that has sampled as deep as 1,500 meters. So my next research question was, are there ontogenic growth patterns in heteropod depth distributions? So the total length of each heteropod was compared by depth range to analyze possible ontogenic habitat shifts. Placenta and Coronata here on the right showed evidence of an upward ontogenic growth shift. And these two species were the largest species caught in the upper 600 meters, and placenta's largest were captured only 200 meters and higher. The other three species did not show apparent ontogeny linked to depth or body length. Previous studies of heteropod ontogeny really has been limited to specific anatomical developments and not really an evaluation of a habitat shift per se. The largest placentas that were bigger than 300 millimeters in length were found only above 200 meters in this study, and the largest coronatas of 150 millimeters and higher were found at 600 meters and higher. This pattern is suggesting that these species are avoiding predation while juveniles, while they're small and vulnerable, and they're moving down into deeper, darker waters during the day. While other species, they didn't indicate ontogenic growth patterns regarding body length compared to depth, there's probably other methods and measurements that should be considered for other investigations that could reveal more on this subject. So let's move to my fourth question. Are the eyes of vertically migrating species larger than the non-migrating? So to see if there were larger eyes found in deep-caught animals, I plotted a calculated ratio from eye diameter size divided by total body length. And I plotted the day and night individuals compared with capture depth by the, per species. And there was not any evidence of large eyes in the deep caught samples. However, we did find a correlation from another angle. I performed an ANCOVA comparison of the diel migrating species to the non-migrating species by total body length and that eye-body ratio. And these trend lines did show that in the migrators, the eye-body ratio is declining with increasing body length. While the non-migrators did not. We calculated the p-value through this ANCOVA that was less than 0.05, indicating that there is a statistical significance between this relationship of the migrators to non-migrators and their eye size and body development. And these eye-body growth trends leads directly into my final and last question, which was, do heteropod eyes develop at the same rate as their overall body or at a different and varied rate? So I plotted total body length compared with eye-body ratios for each of the animals by species. And these three species displayed an allometric growth. This was placenta, Lamarcki, and hippocampus. And these species all have the asymmetrical eyes, if you remember. They're also the smaller eye type. And these species are all the smallest in total body size. I also calculated a linear regression statistic to model the relationship, and there wasn't statistical significance for placenta and Lamarcki. Their p-value was higher than 0.05. These species also had the smallest sample sizes of only 14 and 21 individuals. However, when we did the p-value for hippocampus, there was an indication of significance, and this sample set was much more large at 67. Perhaps the low sample set for the Carinariids affected their statistical outcome here. Coronata and Scutata both showed isometric development trends, and both of these species, again, they have those tubular eyes, the larger eyes, and they're also the largest bodied species. And just like with hippocampus, they also showed statistical significance. And they had a much more robust sample size, 102 of the Coronatas and 129 of the Scutata. And, you know, there doesn't seem to be trends in eye size directly in relation to depth for these heteropods, but that's not completely surprising. Remember, they do mainly occupy that upper 600 meters of the water column, and they're mainly found in the upper 200. That's their best abundance. Although it is interesting, though, that the eye development trends that showed for isometric patterns compared to their total body length, they have those tubular eyes, and they have the largest overall body size, and these are also those Diel migrators as well. So there does seem to appear to be this relationship that exists between the developmental growth patterns and perhaps eye shape that may also be tied into some of the ecological strategies being used by these migrating species. So that was the research that I have conducted over the last 3 years. And just to summarize, we found 5 large heteropod species occupying the Gulf of Mexico pelagic waters. Carinariidae and Scutata are the predominant large species for this region. In these heteropod populations, they're indicating substantial counts, and they're kind of challenging the general notion that heteropod quantities are considered low or in the world. Heteropod assemblages here are including deeper depth profiles than many previous studies, mainly because these have limited their depth ranges to 600 meters and higher. So in general, these deep water collections are really lacking for the world over. One of the most recent studies on heteropod vertical distributions was done out in the Atlantic Ocean by Alice Burridge in 2016, and she evaluated heteropods and pteropods. But again, the heteropods from her study were mainly the smaller atlantids, and her focus was She was focused on the ecological sensitivities of pteropods and heteropods in their ocean acidification effects. We also found that Carinariidae and Scutata are daily migrators from the lower mesopelagic up to the epipelagic, and this is an assumed feeding strategy. These migrators have those isometric growth patterns, have the large tubular eyes, larger bodies, ontogeny in body size and depth was inferred in 2 species, placenta and coronata. Really, continued sampling could reveal more ontogenic shifts among these species because there's a lot of different ways that we could look at those pieces of data. And there appears to be a relationship between eye and body growth in patterns for connection to the eye shape. In Burridge's paper that I mentioned a minute ago, she made a statement, which honestly has been a statement that I have seen in just about every heteropod paper that I've read over the last several years. And all the authors are always compelled to write something like, heteropods are a poorly understood group that needs more focused research, and I am a part of that camp. More heteropod research has a large study spectrum that does completely remain untapped, which reinforces the importance of this study and the need for more research. I believe that this set of data is the most comprehensive, down to 1,500 meters on large heteropod species. And this data is important for future work because heteropods are an important piece that fit into the oceanic food web. They are predators that feed on various zooplankton and larvae, and they in turn are eaten by other heteropods and various fish like tuna and even sea turtles. They're also a part of the deep scattering layer that migrates every day. They're swimming thousands of feet toward the surface for feeding and back down into the dark depths during the day. I said that backwards. They go up at night to feed and back down into the safety of the dark during the day. And I hope that you found this research interesting. I certainly have. I thank you for attending today, listening to the information on these large heteropod species that are found right here in our neighboring Gulf of Mexico. I'd like to acknowledge my major professor, Dr. Heather Judkins at the University of South Florida, St. Pete, for all of her mentorship and encouragement and facilities that I've called home for the last over 3 years. I'd also like to thank my co-advisors, Dr. Brad Seibel and Dr. Mike Vecchione, my committee member, who have really given me such invaluable expertise and guidance throughout this entire process. I also want to thank the University of South Florida, St. Pete, and the College of Marine Science at the University of South Florida just for this opportunity to further my knowledge in my graduate education in marine studies. And I also, in addition, want to thank the Deepen Consortium and the ONSAP program for providing the means and access to these fascinating animals. And this research was made possible by a grant through Gomery, and the data is publicly available through Grid C. And I thank you very much for coming today and listening.